With that, we'll move on to our next speaker, Alexander Buko of Human Metabolome Technologies. And his presentation today is called Novel Technologies for Understanding Metabolomic Communications Between Host and Microbiota. So you can take it away.
Hi, do you see my full screen?
Yes.
Okay. All right, well, thank you for attending the seminar today. I would like to talk to you today about metabolome profiling for these metabolomic studies. As a speaker today, I have a long history of metabolomics, starting with identification of plant and soil metabolites as anti-cancer and antibacterial drugs in the early 80s and 90s, and then up to today where I've been studying with using capillary electrophoresis mass spectrometry, which is I like to talk to you today, and also to express the diversity that we see in the microbiota is reflected in the metabolite diversity when we look at profiling data. Applying our technology to the microbiome is not just a business proposition, but a topic that affects us all and all still holds a lot of mystery. And today we might ask, is there a link between the severity of response to COVID-19 and the gut? The viral receptor ACE2 is highly abundant in the colon and intestinal tract. Could the abundance of this receptor and the regulation of amino acids and other metabolites link to disease severity? Data must be acquired to match changes in the microbiome to clinical phenotype and outcomes to understand the impact of the microbiome, not just on viral infections, but other pathogens and other phenotypes. To reach this goal, advances in pathogen detection and analytics will be required to couple large population studies to bacterial genomes and chemical sequencing. We believe HMT is suited for this challenge. The agenda today is to understand the challenges due to the complexity of the metabolome and to present our technical advantages, to help define these clinical phenotypes and discuss our strategy of metabolomic profiling. Our strategy starts with project planning, then integrating our high-resolution CEMS-based metabolomics data with clinical phenotype and sequencing data, with a common goal to report a library of metabolites or reporter metabolites associated with phenotype and outcomes. To do this, we are prepared to work with any sample type or project size. When discussing the different types of metabolomic projects, the networking of metabolite pathways and the number of metabolites that can be observed, we have to discuss these in terms of the type of sample being investigated. For each sample type, fecal matter, skin, blood, or tissue, each provides a different challenge, and there are common issues that analytical technology must address. To have a comprehensive solution, not only do we need to accurately measure and quantify metabolites, but also to understand where these metabolites come from and to understand their effect on systems biology. Bacteria are in a constant survival mode. They consume and transfer nutrients within their microenvironment, only to be used for energy and biomass production. Leftover materials become waste or potential signaling or transforming agents within the host. Added complexity is that not all the metabolites that we measure are biotransferred from bacteria. There are plenty of endogenous or host metabolites as well, and metabolites from other pathogens. While there are many types of metabolite classes, there are some common types of metabolites that have been observed most often in different studies. These include nucleic acids, energy metabolites, amino acids, and their derivatives, short-chain fatty acids, and oxidized forms of glucose. These common... In 1 second, I'm getting feedback. Can you still hear me?
Yep, we can still hear.
Common types of microbial metabolites are in fact the targets of this unique CEMS methodology. The complexity of the microbiome is defined by the large variety of chemical classes that could be presented in any one sample. However, even when we focus on just small polar metabolites, complexities are still there. Several important metabolites such as phosphate, urea, propionic acid, and butyrates are very small and hard to measure using conventional methods. Many are isobaric with the same exact mass and hard to resolve. Many are highly charged quaternary amines that also can suppress other metabolites coming out through the mixture and separation. As I will show you, the high resolution capability of capillary electrophoresis will allow HMT to measure these small polar metabolites and resolve many isobaric metabolites. At HMT, we use the high resolution capability of capillary electrophoresis to resolve these complex mixtures. We also provide targeted quantitative data at the same time as we produce untargeted data, which is unique in the industry. With exact mass, high resolution separations, and quantitative data, we can then link our data to clinical phenotype, treatment outcomes, N16S RNA sequencing. Many of these polymetabolites are signal molecules that can travel from the gut to the brain and other remote receptors that can interact with our immune system and can be either a benefit to the host, a neuro, renal, or uremic toxin. These signal metabolites are in many cases small polymetabolites and are comprised of many isomers that need resolution if they be used for diagnostic and therapeutic value. Because CEMS can resolve isomers, it is important to understand the difference between significances between functional, positional, skeletal, and geometric isomers. The metabolome contains over 1,000 metabolites under mass 800, of which there are many metabolites with the same elemental formula. The resolution of NMR and in many LCMS methods fail to resolve these isomers, hence lack of specificity for many cases that CEMS can measure. These redundant metabolites exist as functional isomers, such as alcohols versus acids, positional isomers differing by position or substitution, skeletal isomers differing by special arrangements, and cis and trans geometric isomers. To overcome this complexity of size, number, and redundancy, HMT has more than one tool. We have currently 5 different options using CMS to identify polymetabolites and resolve many of the smaller isobaric metabolites. We also have 2 different LCMS options to address the less polar, more hydrophobic lipid metabolites. Let's take a look at several examples of how the high-resolution capabilities of HMT can enhance our understanding of the microbiome. These 2 good examples start with our hexose phosphates, these in green. These glycolytic intermediates are important in understanding glucose metabolism and energy production. With CEMS, we resolve and quantitate all 3 of these hexose isomers. Similarly, we can resolve and quantitate pentose phosphate pathway intermediates, those in red, ribose 5-phosphate and ribulose 5-phosphate. These are critical for understanding the oxidative and non-oxidative pathways of nucleic acid turnover and synthesis. Here we have 8 metabolites, all with the same exact mass and same formula. However, these are a mixture of both skeletal and functional isomers. These isomers are all observed in plasma and can be resolved, measured, and quantitated using CEMS. These small amino acid derivatives and small fatty acid derivatives are of special interest to those studying the microbiome. Here are several small amino metabolites that include branched and straight-chain skeletal isomers, all resolved by CE. In many cases, we see differences in expression of these metabolites and isomers depending on changes in the microbiome in case of obesity, diabetes, and cancer. The ability of CEMS to identify these skeletal isomers is key to understanding the biology and creating the correct diagnostic biomarker. In addition to skeletal isomers, cis and trans geometric isomers can also be resolved. Here, cis and trans C4 unsaturated small chain fatty acids, malic acid, and fumaric acid are not well separated. These metabolites have different pathways and biologies. While both malic and fumaric are endogenous, malic is more water-soluble, thought to be more algal and plant metabolite, while fumaric acid is a TCA cycle intermediate associated with cancer, hypoxia, IBS, and RA. On the right side, trans-hydroxyproline is a biomarker for collagen turnover and tissue degradation, while the cis form is high in grain, oats, wheat, and rye. Hence, these cis forms may be of higher interest in studying the gut microbiome and influences of the diet. Aside from examples of amino acids, small chain fatty acids, the CEMS technology allows for measurements and quantitation of very small metabolites under 100 mass, such as urea, ethanolamine, propionic acid, and many others. As an example of using the specificity of CEMS in this publication, we're looking at the arginine supplementation to promote enhanced learning, memory, and life in a mouse model. For this study, several metabolites track with arginine and polyamine expression, including those 4 highlighted in blue. These 4 small fatty acid amino acids exist as isomers. These 4 metabolites have a combined redundancy of 13 different metabolites. However, the high resolution of CE allows HMT to specifically measure these exact metabolites and to observe them as they trend with arginine and polyamines to create a deeper understanding of the biologies. We see here a direct correlation between polyamine expression, probiotics, arginine supplementation, and expression of the neurotransmitter GABA and the GABA agonist 5-amino valerate, which is a microbial metabolite. HMT has been a provider of metabolomics for over 17 years. As we grow, we add new metabolites to our polar library, allowing us to add new metabolites and develop new pathways. We continue to build our libraries and in-house knowledge base as an extension of enabling and innovative CEM solutions. Many of these metabolites that are observed are reported metabolites that can be connected to clinical phenotype. The following metabolites on the next slides are not solely from our studies, but composites of our observations with that observed in the literature. Hence what I call guilty by association. These metabolites allow us to improve our interpretations of metabolome profiles by connecting metabolites often associated with a clinical outcome. Here are metabolites that can be related to malodor or inflammation or skin sensitivity phenotypes. Many are polyamines and acids that stimulate inflammatory signaling, provide malodor caused by microbiota. Changes in the microbiome can also be associated with hypoxia, changes in oxygen consumption or flux. These glycolytic end products, lactic acid and pruvic acid, are often measured and related to specific bacteria, the accumulation of which can lead to acidosis, osmotic stress, and other local pathologies. One of the most talked about pathways of the microbiome is the relationship of bacterial metabolites to trigger an immune response. Often these metabolites can be related to large unsaturated fatty acids, or to tryptophan metabolites, or to AHR indole receptors. CMS is especially sensitive to detecting these types of small polar metabolites. Taurine and hypotaurine are also related to inflammatory response. Osmotic stress and osmosensoring are common pathways associated with many clinical phenotypes involved in the bacterial microbiota and key stress response. Osmolytes include many types of charged metabolites that compensate for changes in osmotic pressure. These include commonly observed betaine and TMAO to amino acids and polyols like sucrose and glucose. Skin sensitivities and pain can be related to many chemical types: short-chain fatty acids, tryptophan metabolites, and neurotransmitters that are linked to specific receptors. Note that while serotonin is often discussed as a neurotransmitter in the brain, the majority of serotonin is actually generated in the GI tract and is also a plant metabolite. Uric acid accumulation is also associated with local itching and pain. Several metabolites at very high levels can be toxic to the host and can be organ-specific. Kidney and liver toxins cover a wide range of metabolites, including choline, TMNO, hippic acid, and all of which can be present in high concentrations. With our CEMS platforms, we can target and quantitate these toxins by collecting a large number of on-target metabolites. This unique ability allows us to cover a wide metabolome space, quantitate specific metabolites, and discover novel compounds in a single analysis. Short-chain and short-chain branch-chain fatty acids have also been associated with the microbiome for decades. After getting absorbed in blood, short-chain fatty acids can suppress oxidative stress by increasing superoxide dismutase catalyst and reduce glutathione, or by decreasing nitric oxide and reactive oxygen species. These changes induced by short-chain fatty acids can lead to the decline of renal fibrosis and renal tubular damage in cases of chronic kidney disease. However, short-chain fatty acids could also increase Th17 and Th1 cells to promote acetate-induced renal disease. Let's look at these small-chain fatty acids a little closer. I was showing you that CEMS can provide, resolve, and measure these species, starting with propionic acid, butyric acids, and valerate acids. Short-chain fatty acids have one of the longest-running stories associated with bacterial metabolites, along with lactic acid. While these short-chain fatty acids have been related to many pathways and signaling implications, they can be linked to specific receptors and are subject to a growing list of ongoing studies. However, the ability to resolve, measure, and quantitate this diversity has been very difficult and challenging. The initial interest in short-chain fatty acids began shortly before 1971 and peaked around 1975, as seen here in green, through using a PubMed search for short-chain fatty acids. The ability to measure these, however, was limited in time to thin-film chromatography, highlighted here in red, which did not have either the sensitivity or resolution to resolve and quantitate them. Hence, these studies slowed down for decades until NMR and LCMS methods began to do a better job in the 1990s, and interest started to shift into the year 2000. About the beginning of metabolomics, while an increase in shifts again due to other technologies such as 16RS sequencing and CEMS. Today, we collect more accurate representation of the microbiome using genetic sequencing and a more accurate representation of the microbiome using CEMS as current enabling technologies. In conclusion, we have seen that the metabolome profiling represents changes in the bacterial composition. Such profiling can be composed of a complex metabolome space of charged and uncharged metabolites. That for many cases of small polar metabolites, their best detection is measured by using CEMS for specificity and selectivity, so a direct connection can be made to clinical phenotype and biochemical pathway. Before taking questions today, I'd like to take a few words about our company, HNT, and something about our CEMS technology. We opened our doors in 2003 in Japan at the beginning of the metabolomics revolution. Then in 2013, we opened an office in Boston. In 2018, an office in Leiden. As mentioned, our CEMS is a different type of separation. In CMS, metabolite separation is quite different from liquid chromatography. An uncoated open fused silica capillary column is used to separate metabolites using a 25,000 volt electrical field to migrate and separate metabolites based on size, polarity, and shape, affording us a precise resolution that we need to separate isomers and to measure small molecules. HMT has patents and publications covering the interfacing, hardware, and software programming innovations needed to bring this technology to you. We have discussed the complexity of the microbiome. At HMT, we have different options so we can cover a large chemical space, from 2 different LCM methods using MScan and DualScan to cover long-chain fatty acid and acylcarnitines, to 5 different CMS methods based on using basic, advanced, and omega scan for looking at small polar metabolites like short-chain fatty acids, amino acids, and nucleic acids. We also authored a quantitative analysis using C-scope to cover glycolytic energy pathway and the F-scope to cover measuring isotopomers for using labeled glucose or glutamine. Here's a summary of our different hydrophilic, lipophilic, and quantitative options. With each client, our scientific liaison will work with you to choose the right platform or combination of platforms to suit your needs. Each project includes all statistical and biological interpretations with a fast turnaround time. In conclusion, we have demonstrated that the complexity of the microbiome requires enabling technologies that can resolve, measure, and quantitate many chemical families. At HMT, we're using accurate and comprehensive data sets using both CEMS and LCMS to link metabolic profiles to clinical and disease phenotypes to specific bacterial profiles to create diagnostics for drug in the health industry. At HMT, we're the only CRO to provide CEMS profiling and the only CRO to produce both targeted and untargeted solutions from the same analysis. We use this technology to better measure the metabolome and to be your collaborator partner with other 7 different CE and LC options. Thank you for listening, and now I can take questions.
I think we have one question here. How many metabolites in total can you detect?
Well, it depends on the type of sample. We've looked at sweat off the skin. We've looked at Alcaris plasma, the normal fecal matter. So it really depends. So it could be, only a few hundred in something like tears and sweat, or it could be 700, 800 using both our CE and LC options, looking at things like fecal matter and plasma.
Thank you so much.